we're going to switch gears a little bit for a panel discussing the future and path of AI and biotech and the life sciences. And this is a category and area that Madrona has been investing in really for the last 5 or 6 years. Talked about it a little bit earlier in the day as we were talking through the funds, and a space that we're super excited about. So I'm thrilled to have 3 of our founders and CEOs in this space here. And since it's always hard to do it justice, I'm going to let them give really brief intros of who they are and what they're building, and we'll jump right in. So maybe Jim, since you're right there, I'll start with you.
Sounds good. Thank you, Chris, for having me and the Madrona team. Nice to see everyone here. My name is Jim Martineau, co-founder and CEO of Gnosis Bio. We're a AI-enabled biotech focused on cell-targeted RNA therapeutics, which is a very exciting modality for the treatment of major chronic diseases. My company is focused internally on immunology and inflammatory diseases, but we also collaborate with some of the largest pharma and patient advocacy groups in the world, including Johnson & Johnson, Daiichi Sankyo, and the Cystic Fibrosis Foundation. And with Madrona, we've been working with you guys for over 3 years, so excited to be here and talk about what we're doing. So thanks.
Thanks, Jim. Jamie?
Hi everybody. Name's Jamie Lazarevitz, CEO and co-founder of Archon Biosciences. We are a generative protein design company, and I don't know how many people here from the Seattle area, but protein design's a pretty hot space right now. But come from the Institute for Protein Design, where we developed this technology over the last decade, partnered with Madrona about 2 years ago, have now built our team to about 20 people. And we use generative protein design to really change the way we design drugs. Antibodies are this fantastic class of molecule that we use to cure and treat problems such as cancer, and we're able to change how they behave inside of the body to make both more potent and safer medicines.
Thanks, Jamie. Jeff?
Hi, everybody. Thanks very much for the invitation. My name is Jeff. I have a couple of jobs and interact with Madrona a lot on all of them. So I'm the Chief Data Officer and a professor at Fred Hutch Cancer Center here in town. And through that have started this Cancer AI Alliance with Matt McElwain and many others. That is bringing cancer centers together to integrate our data through federated privacy-preserving learning. I am also co-founder and CEO of a Madrona-backed company named Synthesize Bio that develops foundation models to generate experimental data from AI models instead of having people pipette things in a lab. So I have the 2 hats that I'm kind of representing here today.
Thanks, Jeff. Well, we're super excited to have you all here. And one of the things I think that is obviously becoming quite a hot term out there is this convergence of AI and biotech. Sometimes it's called tech bio. And I think as we were discussing with this panel, we wanted to start with sort of the definition, which is, I think a lot of people use the phrase, very few people say actually what they mean when they're saying AI bio or tech bio companies are using AI. So I thought we'd start with everybody's individual view on what does that mean? How do you apply AI and bio? And Jeff, maybe we start with you for sort of the 10,000-foot definition, and we can dive in a little bit more with Jamie and Jim.
Yeah, sounds good. I think for here, the sort of perspective that we like to use for AI at the Hutch, for example, is AI and biotech. It gets applied, I think, both tech bio, biotech. Sometimes it's used as a direct tool to make a prediction. Sometimes it's used to generate new types of molecules to pursue. But the 3 components we always think about are, for any AI, are what data underlies it, what data have we collected that's unique, original, new, what are the models that enable us to do things with that data that people haven't been able to do before. In the protein space, there's been a huge amount of development there. And then what are the interfaces, the ways that those models translate into action? And I think at that interface is where you see a lot of difference between tech bio and biotech. Is it moving a drug forward, or is it a piece of software that people use in the process? It's sort of what is the interface to the rest of the world is the kind of place where I see a lot of that difference happening.
Jamie, you were at the IPD for a long time, and the IPD, which is David Baker's lab here in town, has certainly been on the forefront, along with Google and AlphaFold, of producing a lot of these models. But I'm curious, when you think about putting AI into sort of the applications that you're using, how do you see that intersecting with sort of the biology and the traditional biotech approach?
Well, I think the interesting part is that before we were separating this notion of tech bio and biotech, but the reality is as these models and these applications become better and better, fundamentally you're at a point where you actually can't separate them. So any biotech company that is not using artificial intelligence, is not using these machine learning methods, cannot make the types of insights or products that a tech bio company can. So I think the big thing is because things are becoming so sophisticated, one can't just exist without them together.
And Jim, at Gnosis, you guys have done something maybe a little bit different, which is you've built your own model. And that's certainly a slightly different approach than people who've maybe pulled in different parts of open source models or refined them. And so maybe talk a little bit about why you made that choice and how you think about that as an advantage, and certainly how Gnosis is going about building different products.
Yeah, absolutely. So when we were starting Gnosis, we spent a lot of time thinking about the right application of AI in bio. It's a little different than consumer or enterprise, where faster, better, cheaper creates value very quickly. In bio, you have really long product development life cycles, and the stakes are really high. It's not enough to just apply a cheap model to do something we're already doing. We really have to unlock new capabilities. So we spent a lot of time talking with pharma executives about really where the challenges are today in the traditional pharma biotech industry. It comes down to two key challenges that we've solved with our model development. The first is the challenge of safety or specificity of medicines today. The canonical example is chemotherapy, which is life-saving for some cancer patients, but is very well known to cause a lot of negative side effects based on how it impacts really all growing and dividing cells in your body. And if we had a chemotherapy equivalent that was much more targeted and specific just to a tumor cell, for example, we'd be much more excited about that. And then the second challenge that pharma faces right now is massive problems in innovation, loss of exclusivity of many of the products that have generated most of the profit over the last couple decades. And so when pharma executives are thinking about what's next, they're saying, how do I access the next set of targets, the next set of biology? And I need the tools and modalities that allow me to go beyond what's been possible before from traditional medicine development. So we took efforts to build in those specific areas.
I want to ask a question about sort of the two extremes of this world, and I'll let any of you jump in and answer it first. I think to paint the extreme versions of the view, you have one view which is AI can do everything. And you pull in a model, and on the other end, a drug will come out of it. And on the other end, you have AI can't really do any of that, and so you should just hire a bunch of scientists and do things the way you've always done them. The truth probably lies in the middle, but I'm curious where you guys all think we are on that spectrum. Jeff?
Yeah, I'm happy to jump in, but if others have opinions. I would say, given that I'm spending a lot of my time doing AI, I'm a AI hometown crowd person, but I would say that for me, the big transition, I used to be an AI skeptic in a big way, and I think the big transition for me was not thinking of AI as something that replaces humans, but rather it's a better computer for humans, has been a very useful reframing of the problem, especially in my role at the Hutch, where we deal with medicine a lot and healthcare systems. And there was a lot of hype for a long time about we're not going to need radiologists anymore. Like there's going to be no radiologists in North America. If you don't know, there's a radiologist shortage in North America. If you know any radiologists, you should introduce them to me. And so what we've seen is though that the radiologists that are being trained and coming out of residency and fellowship programs increasingly have to know how to use AI tools to be successful in their job. So it's not that we lost the need for radiologists, it's that being a radiologist means something different with the better computer that is AI. That's kind of how I think about it.
And Jamie, there's certainly a bunch of claims out there that all you need is AlphaFold. And you can tell AlphaFold or maybe some of these next gen models what to do, and you'll get the right protein out of it, and that's sufficient. What's your view on that? And how do you sort of merge those models with the actual task of creating next generation proteins?
Yeah, so I think we're certainly at a point where we would like to term the field of protein design as we're in a space of post-evolutionary biology. So the ability to make proteins has never been easier. But that doesn't necessarily mean that the things that you make are useful. And I think that there's this big divergence between these two things where it's just because you can make something doesn't mean that somebody wants it or somebody's going to use it. So there's a lot of nuance. And so when you're applying these things, because biology and medicine is so multivariate, so multifactorial, just because you can make something doesn't inherently make it something that will be good and work well in the body. And so part of it is tempering down claims, but also knowing that you can now make things that are Again, in the sense of post-evolutionary biology, you can make things that you could have never evolved, you could have never made in the first place. So it's both something that is like wholly enabling, but also one must like temper expectations to put it through the necessary trials as required to make it into something real.
Great. I'm going to skip around to one of the questions just because we sort of mentioned it, but there's also a conception out there. Google just released their co-scientist model. I'm not sure how many people saw that, but it's a fascinating way to have sort of an autonomous research agent. It's super cool. We talked a little bit, I know Satya mentioned a little bit about the code gen earlier during his talk around how it's just incredible to see that now some of these LLMs can produce really good code. Maybe the provocative question is AI going to replace scientists? Are these tools getting powerful and strong enough that a lot of this work can be done by AI, or what will that look like in the future?
Yeah, I just finished performance reviews for my team, and I get this question about 10 times a day. Luckily, the answer is, for right now, it's no. What's been really interesting about AI, and I'm sure you all see this in your personal lives and work as well, is it's really surprising. The big thing that's happening is all of our team is spending less time at the computer. Any task that was a computer task just takes less time. Or drug design, or molecular optimization, writing, policy, procedure documents, protocols, what have you, is going rapidly to zero. But in building a biotech company, building medicines for patients, there's an enormity of lab work. So we're in the lab, we're developing assays, we're validating those, we're doing work in a vivarium, and that work we're just able to spend more time on that, which is really the hard, critical work in building a company like this.
It's interesting because I would almost want some of our employees to spend more time at the computer. We rely so much on automation right now. We use a lot of robotics in what we do from a day-to-day standpoint, that if you're a scientist and you don't know how to use AI, you don't actually know how to code, then it actually becomes very difficult because your throughput is so much less than someone that is able to interface with some type of robot, for example.
Jeff, we used to joke about the sort of question of, is it easier to teach a scientist how to code or an engineer or a coder science? I think we've talked about that a lot. So I'll ask you a slightly different version of the question, which is if you put your professor hat on and you look at the talent that's emerging through undergraduate programs, PhD programs, postdoc programs, what does that look like now? What are sort of newly graduated PhDs and postdocs coming at the world with in terms of skill set on both the engineering AI and the science side?
Yeah, I mean, the thing that's, maybe I'll go even scarier than that. First of all, I think that answer is now kind of definitively like trending in one direction. I have terrible hands in the lab, and like I can, now with the, coding assists and vibe coding and other things, like people can figure out how to code much faster than they used to, but having good hands in the lab is still a pretty tough talent to acquire. I'll give you a scarier answer though. I have a 13-year-old son. He is currently engaged in a project where he's developing a reinforcement learning solution to his favorite board game with PyTorch, largely assisted by these online LLM type coding tools. He's 13, and so if that's a 13-year-old, you can imagine what the kids that hit undergrad and grad school look like. I think we're seeing an incredible acceleration in the talent, especially in the computational and AI side of the space, and what that's going to mean for all of our industries, I think, is going to be really interesting to see.
Yeah, I think, just from my perspective, as we talk with, these three and our other company leadership around recruiting talent into their companies, it is fascinating how it has gone, I think, a lot from a lot of pure science to whether they had it or taught themselves it. People are very familiar with using these AI tools, and not just using them, but deeply modifying them, fine-tuning them, some of the points you were making earlier. It's pretty impressive to see the pace of evolution and sort of the talent in this world. But I want to go back to sort of the business model side of this, which is as you think about what should a biotech do with the AI tools at their disposal? I don't know that's an easy question to answer. I think the views can range from what we call sort of a platform approach, which is you build a lot of infrastructure and discovery and software, and you make lots of potential therapeutic assets and partnerships, to a very single product, go make one drug really well. How do you guys think about platform versus product?
Probably makes sense for you guys to.
Cool. I'll take a first stab at this for Gnosis. So we are both a platform and product company, and The way we think about it, we're here, we all left what we were doing to do this, to build medicines for patients, and the only way to call success is, we've actually done that. We've taken a medicine all the way, and it's now available, and it's helping someone in need. So it's, in our view, it's necessary. It's the only measure of success, that you go the distance in building a real product for a patient. That being said, the way we build, we think a lot about how we solve problems. Are we solving it in a repeatable, reproducible fashion so that we can very easily plug that exact same platform data set methodology into the next target, the next pathway, the next disease indication. Over time, we imagine that the best platform companies are those that focus on building one product sequentially as the company builds.
And Jim, maybe I'll ask you just a double-click question on that, because you just announced 2 new partnerships with pharma, which I think is just fantastic and is a great achievement for Gnosis. And sometimes we hear you should do partnerships and you should generate revenue, and sometimes we hear, nope, you should just take all of that and look inward. But you chose to go down the partnership route, and that's been successful. So how do you think about integrating that sort of revenue generating function into your business model?
Yeah, absolutely. I think there's a lot of art in how you partner in this space. It was very important for us to find great partners that have very deep expertise in their biology. One of our partners is Johnson & Johnson. They've been working for about a decade in the area that we're collaborating in. Bring enormous expertise to the development of this medicine, and essentially had a missing piece, which is a platform we've already developed. And so for us, we're essentially plugging in a technology we've already built, and they can take it forward and build medicines on that. So it's very low opportunity cost for us, and we work with them and we share in the risk upside as the product is built. I think a lot of biotechs, they can overcommit themselves to partnerships that take a substantial amount of their time and energy, or really take away the opportunity set for the company. So we've tried very hard not to do that.
And Jamie, maybe from your platform approach, I wish we had a visual, because it's kind of fun to see what the Archon molecules look like, but you're building molecules and potential drugs that basically nobody has ever seen before. And so as you think about progressing those, and by the way, that were not possible, right, without AI and protein design, techniques developed here at the IPD. But as you think about that, and you talk to partners, and you think about what it takes to push those forward, are you encountering sort of any unique challenges, or what are you hearing from the market as you sort of push biology into kind of an unknown direction?
Well, I think the big part is being able to frame the problem that you're solving. And so if you're able to clearly define what that problem is, as long as it's not too far off center of speaking to these groups and these people, then they're able to start thinking and empathizing about actually how they could integrate it into their workflow. I think the big part is that when you're doing something that's totally different, something that nobody's ever really seen before, then the bar just becomes a lot higher. And so therefore, in order to prove that it is something that people have to adopt, you have to be really, really strategic, really, really thorough about proving that you're able to overcome these limitations that all other technologies could not solve before. So it is a big education piece. And unfortunately, and I guess fortunately, you have to prove that through data.
And Jeff, we were talking about this before. If you put your Synthesize hat on among your many hats, Synthesize is not focused on developing drugs, unlike Archon and Gnosis. And so that has a whole different set of potential business model implications and how you think about putting your product in front of customers. And so as you have built Synthesize, what are the differences there? And when you go talk to potential users of your models and your software, what are those conversations like? What are people asking? How do you think about that in that framework?
Yeah, I think the simplest way to describe it is Synthesize Bio is a data company, like many companies that have aggregated and put data together, which then is a product that people use that data to make discoveries, to move their molecules forward, to move their products forward. The difference is we don't have people in a lab pipetting, we have models that generate that data. And so it's data is data, but it's generated in a different way, in a faster way. And so we've seen applications ranging from VCs who want to vet new opportunities for investment with data from our models, to cohort generation for large-scale studies, to toxicogenomic studies for molecules that we haven't seen before, like how do these models actually generate tox studies rapidly for a new material that we haven't seen before. So we're seeing this really wide range of potential applications, but they're all centered around people need data to answer the questions, to back up the assertions that they're making, to educate their customers about what they need to do next.
And maybe going a little deeper on that, you can put any of your hats on to answer this question. But as we all talk about sort of the model side and the model quality being related to the data quality, we hear all the time a lot of the good data, right, is not in the public domain. So unlike the ChatGPTs or the large language models of the world, you can't really scrape the internet to get protein data, small molecule data. There's some out there. It's maybe not great. And so how do you think about access to that data and sort of the proprietary side of the data and/or pulling in enough to make these models accurate and work for those functions?
Yeah, and I'd certainly be interested in Jim and James' perspective on this as well. On the synthesized side, we both have proprietary and public data that we integrate in large quantities to be able to generate our models. Through Kaya, this Cancer AI Alliance, we're trying to integrate the electronic health record data of multiple academic medical centers, which is a huge, huge challenge for getting access to a type of data that has historically really only been available through companies like Flatiron and Truveta for community-based practices. So regardless of how you go about it, like sort of the lifeblood of AI platforms is what data can you generate and can you develop to build these models. And so some companies, I think, are pursuing developing their own proprietary data, some leverage public information, but at the end of the day, the data is sort of the lifeblood of these models. So I'd be curious what.
Yeah, I mean, I think that's why it's difficult to get your hands on it, because people do know it's of value. And so if they can figure out how they can monetize it themselves, then it also de-incentivizes them to want to share it. So there is this balance between the two.
Yeah.
On the other side, I would just point out that the cost of data generation is dropping very, very rapidly. Yeah. So right now, a couple years ago, we're looking at 10,000 cell data sets of RNA-seq. Now we're looking at hundreds, thousands, and just recently we're up to 100 million. And those are all, in general, being released publicly. And so I think as we go forward, there will just be more and more data that's available about how our biology functions, and we're able to use that very actively as we build our programs.
Is there a magic amount of in-house proprietary data you think you need to generate for your models to be at the quality where you want them to? Jamie, Jim, maybe either of you can take this. You both generate a lot of in-house proprietary data. What's enough? We certainly hear lots of big numbers for some models, and where do you see that as sort of the scale of data generation? Is it really truly improving the model outputs?
Well, this is also the balance between product and platform. So in like the pithy way that we call ourselves a product-led platform, is it's hard to know a priori what enough is. And so you really have to develop a product or develop an asset that is actually shown to be that much better than anything else that's on the market. And it's kind of through that process that hopefully you've learned how to make a better molecule, how to make a better drug, and that the system to do that, you have this fast follow. And so I think you actually have to go through the process to quite a late stage before you truly know that what you're making is actually of value. And I think that's the problem with a lot of these AI drug development companies, is that fundamentally we find that you build these big data libraries, but in actuality, the data that you've collected doesn't allow you to generate viable molecules. We're not seeing as much translation as what was touted 10 years ago, because you really have to focus on the product first and then figure out how to build it out after.
I fully agree. I mean, it's a huge problem in biotech. We have so much data that's been generated that's essentially useless for therapeutic development. It's really sad, honestly. So the way we approach it sounds very similar. We think about all data generation is product directed, and really think about it as kind of a reinforcement loop around optimizing a given product. So we'll start with public data or what have you to build a first generation of our product, and then we start to generate data around that, and we are optimizing our molecule using a bunch of signals from the lab, from our model predictions and other physiochemical structural feedback to optimize. And what we've seen is that when we go and we find another big pot of data somewhere online and we put it in to our system, it doesn't add signal. It was kind of wasted. So I do think I wish more biotech was focused on the product-led platform building.
But if you add proprietary data you generate, does it add signal to the model?
If it's product directed, absolutely, yes.
Got it. I'm going to try to ask a maybe controversial question. I don't know. We'll see if we can get a little bit of disagreement. Obviously, everybody is excited about the various approaches of AI plus bio here. But if you think about the design problems that these AI models are being pointed against, obviously here in Seattle, we're it's certainly a world center of excellence, maybe the world center of excellence in protein design. But there are other problems out there. There's small molecule design, there's very large molecule design, there's oligo, which is basically DNA, RNA design, there's cell therapy design, and AI is being applied against all of these different approaches. Do you think it's going to solve one of them first? Or which one is the easiest problem and which one's the hardest problem?
Well, we're an RNA therapeutics company, so you might guess my answer, but I guess solve is such an esoteric question because there are so many models that get published every day and beating some benchmark. But is that helping a patient? The way I really think about solved is what's the impact to the industry overall as we look 5 to 10 years out. And as we think about it, we're such a product-led company. We spend all our time talking to pharma. And again, the big problems pharma is facing are the safety and the lack of new target access to new targets. The RNA space is really ripe, and we think this is the right time for that modality to really grow. And we think AI is really the key ingredient that unlocks the modality. So my prediction, you look 10, 15 years out, RNA therapeutics have been enabled the most by AI.
Jamie, Jeff, do you have other opinions?
I mean, I think protein therapeutics will be the most enabled, but
this wasn't my plan to try to get you to just answer this.
But again, what's interesting is the same reasons why you think that you're going to have impact is the exact same reasons why we think we'll have impact. If you rely on natural molecules to solve problems that they never evolved to have in the first place, they have obvious dysfunctions. If you think of side effects or toxicity, these are natural consequences of how these molecules either evolved or just their inherent behavior inside of the body. And because we never had the tools to begin with to be able to engineer a drug versus co-opt something natural, we weren't able to overcome these issues. But now we're at a point where if you look at distribution, if you look at toxicity, if you look at efficacy, I do believe that these are the low-hanging fruit applications in targets that people really care about that were limited by completely unnecessary side effects. So I think the types of problems that you're talking about are the best problems that are accessible to be solved. But it's going to be solved in a variety of different modalities because they all suffer from pretty similar problems, even though they're structurally different.
I mean, my lab studies gene expression and RNA and molecules, so I have an obvious horse in the race too. But I would say that the interesting thing for me is thinking about, when you say solve, do you mean solve? Within the protein space, there's some definitions of solve that things have already happened, right? You can take sequence and predict structure in ways that you couldn't just not very long ago. So it's already solved by some definition. But speaking to what I really love about what both James and Jim have said today is product-led sort of development. The thing that's interesting to me is once you have a new product that you're considering or that you want to sort of push forward in a program, there's a certain amount of irreducible time it takes to do that work. All the experiments you have to do from the moment you have that new product to it's actually on the market and patients are able to get that product. And for me, solve, the thing that we're focusing on with synthesized bio is how much shorter can we make that time by reducing the time and cost of doing the experiments that we take to go to the sort of end stage. That's obviously a long-term vision for how this works, but it goes back to your agentic sort of thinking is like in the future, is this a bunch of labs connected together? Is it a bunch of agents with a bunch of synthetic data generating models sort of pursuing that at a much faster speed? Because right now there's an irreducible amount of time, no matter how much you solve a molecule at an early stage, you're looking at a very long development cycle.
Yeah. So. Well, I think it's because it's so fragmented. You have everybody doing the same type of work. Nobody's incentivized to share their data because once you move beyond just the basic cell interactions, it's the cost to acquire that data increases and increases, and the value of it to what you're developing increases and increases. And so there's no reason why you would want to share it.
Yeah. Well, I see the clock hitting 0, and so that seems like a pretty a good place to stop. But I really want to thank all three of you again for joining us here and talking about this topic. And I think we should be really excited about the future of where this intersection is going. So thank you so much.